I will now switch to our next presenter, who's from Ecuador, and that's Professor Hernan Morales. He's a physicist. Since 2024, he's a research professor at the Universidad de las Américas in Quito. He did his PhD at the Max Planck Institute of Molecular Biology and Genetics in Dresden, works on machine learning applications and computational models for biological systems, with a focus on embryonic development and tissue analysis. Today, he will share his vision on global open source platform where researchers share embryonic imaging data and get in return ready-to-use AI analytical models. And I'm sure you will tell us more about it.
First, thanks for the nice introduction, and thanks to the jury and to the organizers for inviting me to present my project. So let me quickly, I will share my screen. So today I will talk about the initiative that we proposed that is called Image2Model Exchange. That is a way to democratize AI analysis through fair data and model sharing. So we are living in the era of bioimage analysis and the AI-powered era of bioimage analysis. Actually, AI is everywhere nowadays, but I will focus on bioimage analysis. And I'm going to show you a few examples, like how we can use these methods to increase, for example, the quality of the images. Here we have images of nuclei increased, but also to reveal information that is not visible by human eyes, like in these bright field images where you can do virtual staining, meaning you can visualize structures that are not visible directly from the microscopy, but you can also use these methods to extract information, like through segmentations where you can reconstruct digital objects and get quantitative data, which is very important in biological science, right? To extract not only seeing the data, but also extract information. And I want to focus a bit in a more narrow topic, that is the computational embryology, that is relatively new. So this was recognized as part of the Method of the Year in 2023 by Nature Methods. And here we have the opportunity to contribute with two works. So the first one that is called Embryonic. Embryonic is a deep learning method that basically takes videos of images, very simple images, bright field microscopy images, the easiest ones that you can take, and identified different phenotypes caused by mutations. So this is how it works in practice. So you can detect the embryo and then tell what is the mutation that is there. And other part of our work was focused on a very big problem that's also in developmental biology labs, that is to identify the stage of the embryo. What is the stage of the embryo is like the morphological age of the embryo, in which stage if you have the 2 cells, 4 cells, gastrula, or you have the formation area of the body. So we also develop a deep learning method that then does this in an automatic way. So removing the bias and making it way faster. As you can see here, I show you a lot of different cool tools that can do many things that were not possible before the era of the deep learning, and now they can help to the researchers to do their job in a faster way. But with great power also comes great responsibility and big challenges. And I want for you to have a clear idea from where the challenges are coming. I will quickly explain how the AI model is trained. So initially you have data, and then for deep learning models, you need a lot of data, and also you need annotations, meaning what you want to get out of the data. On the other hand, you have an AI model that you can think about it as a black box, something that the computer is doing that needs to learn a task. And then it comes the important thing, that is the training. So you give the data to your model, so the model learns what you want to get. And then finally, when this process is done, you have your A-trained model. So you can imagine, as everybody has used ChatGPT. So the ChatGPT is already a trained model that you give the input, so new data, and then you give an output. So you are not involved in the process of the training, just the final user. So, but what is the problem here? So probably you have seen this in many papers along the years, not only with AI, is that data is not available, code is not available. And then we suffer this here as well. So data description is almost there, always there, but data for the training is almost never shared. So models, description of the models and code are often there, also published, but the reproducibility is not there either. And when we talk about trained models, so the final output, the model that you could use, sometimes is shared, sometimes that is not even shared, but the evaluation data is very rare. So I think you can see that there is a big challenge here. And then I wanted to formulate a bit more and then talk about developmental biology. And what are the problems here? So there is a lot of data fragmentation. There are millions of images of embryo taking all over the world nowadays, but they lack standardization. So we cannot reduce it, or they are just not shared openly. So nobody can use it over the world. So there are also access barriers. Most of the computational power and the expertise to do this is the global north. As scientists that have been in Germany and come back to the global south, I can, in the first, can tell you about what are the limitations of the computational, what are the barriers of not having the computational power and the expertise in the global south. One of the biggest problems now that I see is that there is a lot of valuable data, training models, and code that remain accessible to others. So data is here, models are there, but we cannot reduce it. So, and then the first solution will be like, why we don't just share, right? But sharing is complicated. And I think everybody could agree that especially in earlier stages, sharing your data is something that you at least think twice before doing so. So what we propose is to go back to a method, to something that worked for humanity long ago, that is bartering. What if I don't propose you only share the data, but I give you something in return? I give you a model that you can use to analyze your data, and then you get something in return also, not only sharing your data. So, and we're, so this sounds very utopic, and then but fortunately we're in the shoulders of giants. There are a lot of initiatives for sharing data, that the image data resource, bioimage archive, Zenodo, but also to share models like GitHub, Dockers, or new initiatives like bioimage models too, where you can actually post your data, share your data, following transparent policies. However, we see that there's still a gap between these two worlds. So here's what we propose in Models to Exchange a project, is to create an ecosystem based on reciprocal bartering. So whereas researchers can take your data and get AI models in return. So basically the idea is that we will be a link between data and models. So this is one of the pillars of the project. And how do we propose to do this? By linking people, linking microscopists with computer scientists. So we enforce fair principles and standardized formats for sharing the data and the models. And what is very important here and makes it different from other proposals is open publication. Because at the end, all the address that I said, we can do it and people does it in the daily basis. You talk with a collaborator, the collaborator does model, you share your data, you get the model back, but it stays there. So what we propose here is that we also open up the data and the model so that anybody can use it. So this sounds super big, so we have to do it step by step, and then we go narrow to a simpler problem. This is in my field of expertise, and it's computational embryology, and it goes in 4 steps. So the first one will be a call for data, try to encourage labs to join the project, and we focus on very specific data. So 2D imaging of bright field, fluorescence, and wide-field microscopy, because it is cheap, is super well implemented, and it can be done basically anywhere. So we aim to partner with global consortiums, the Global Bioimaging and all the regional ones, Latin Bioimaging I'm part of, Bioimage North America, African Bioimage Consortium, Aero Bioimaging, but also societies or like developmental biology, or journals that promote open science like eLife and companion biologists. And actually we have already 4 partners willing to start joining the projects. Diana Pineiro, Daniel Chapek in Austria, and then some people in the Global South, Andrea Romero and Fabian Segovia in Chile and Ecuador. So what do we do next? We could do curation and data standardization. This is a hand-in-hand work with the contributors. So we curate the data, checking that everything is there, all the information is there with the correct standards. We follow the community standards, recommend metadata for bioimage analysis, OMSRs, the format that is specialized for standardization bioimage. Then we will load the data in the bioimage archive. Then you get a persistent identifier. And then once we ensure the fair compliance, so we add the information to the database and the website of the Image2Model. So what we do next? So then comes my expertise a bit. So we generate the AI models, and we focus also on very specific tasks, embryo segmentation, abnormality detection, and phenotype recognition. So it's also reduced so that we can do it actually in the time of the project. And finally, we do open publication and release all the code at different levels for developers, in GitHub with all documentation and permissive licenses. If you are an advanced user with containerized environments for reproducibility, and also we will join the initiative of the Image2, where you can put your model that is ready to use for the researchers with all the information. So what is our final goal? We want to unlock discovering of embryonic development. So by gathering community of scientists, we aim to create an open multi-space data embryonic database. So meaning we will have a huge database, and we find that this is one of the most ethical ways to collect data for the computer science community so they can work in AI models and ideally formulate a foundational cross-species AI model for embryonic development that in the latest point will be used again for the biologists to get an unprecedented comparative predictive analysis cross-species. So with this, we open the data, we open the models, everybody can use it, and then we open science. In our long-term goal, we don't want to stay just there, and then we hope that this will be a community-driven process. We have big communities like the Global Bioimages coordinating and promoting this for biologists and microscopists, and the Global Bioimage Analysis Society doing the same for the computer science, and we'll be just the link, the catalysis between these two worlds, trying to put these things together and then try to promote open science. And with this, I just want to thank my students and my collaborators, and thank you for your attention. I'm happy to take any questions.